Hello and welcome. In this video, I'll be talking about the exercises on sequence manipulation. In problem 1, we're supposed to estimate the probability that a random sequence that is 100 bases long and with equal probabilities of A, T, C, or G will be cut by the restriction enzyme EcoR1, which has the recognition sequence GAA-TTC. Provide your answer as a probability between 0 and 1 with two significant figures. So in this problem, we have, if I go up, in this problem, we are given a function that generates random sequences. And if we run it, we can see that a random sequence is made. If we run it again, a different one is made. And the idea is we run this function many times, and there is a chance that we happen to encounter the EcoR1 recognition sequence by random chance. And the idea is to perform many experiments, a large number of experiments, and to find out the approximate frequency that this event occurs. So you can do it like this. We have a counter, cut count equals 0 to begin with, that we're going to increment whenever we find the recognition sequence. We have a total count, which is going to be the number of iterations or experiments that we perform. And we'll set this equal to 200,000, nice large number. And we have a for loop for experiment in the range from 0 to total count. So this will perform 200,000 experiments. And each one of those experiments constitutes generating a random sequence. So you can see here a random sequence variable is made, and it's created using the generate random sequence function that we made earlier. And we simply check with a conditional statement here. If the sequence GAATTC is in random sequence, random seq, then we increment the cut counter, cut count plus 1. And then finally, after this loop is finished, outside of the loop, we simply check what was cut count divided by the total count out of the 200,000 experiments, what fraction of those had the recognition sequence. And it takes some time to run, but this is important because if we use a smaller number of experiments, then we have a chance to, due to random chance, get a different answer each time. And the larger, the greater the number of experiments, the more precise our solution is going to become. And and it will begin to approach the true frequency, the global frequency. And here we get an answer. So 0.0228, which rounds to 0.023. And you'll find that the majority of the time, if you run these experiments, you will get 0.023. But it can be that occasionally, due to random chance, you get something slightly different. But the larger the numbers get, the more we will be approaching this value here, 0.023. Okay, and the next question asks us to fetch the sequence of Phi-29 polymerase and use a sequence to figure out the molecular weight. So we have this DNA sequence here, Phi-29, and we also have some information. We have a list of masses and a list of amino acids. So if we check the type of Phi-29, the DNA sequence, we'll see that it's not a string, but a BioPython object, a sequence object. And this means we can run this translate method on it, which will give us the protein sequence. The other thing we need to do is get a dictionary out of our list of masses and our list of amino acids. And so if we look at this line here, we can see that there's this zip statement here, and zip, all it does, and of course we're copying this from tutorial in the exercise, but just to explain this a bit, zip is going to take two lists and pair each corresponding position together. So element 0 in amino acids would be paired with element 0 in masses, and so forth. And so you can see it pairs them in this way. And so when we feed this to a dictionary function, then it will generate a dictionary which has the keys generated from the first list and the values generated from the second list. And you remember, to use a dictionary, you always use the keys to look up the values in that direction. And we can see how if we call, therefore, an amino acid code, such as S or V, for example, we get a mass. So then to estimate the molecular weight of this protein, we simply translate it, and we can loop through each amino acid in the translated protein sequence. And inside the loop, we'll just be keeping track of the total mass. So we'll use the dictionary to look up each time the appropriate amino acid, because our iterator is taking on the values in that protein sequence. And so it will have values such as V or W or Y and so forth. And then if we feed that into the dictionary, just like we did up here, then that should return us a mass value that we just tack on to our growing sum here. And the first time you try this, you might have encountered the following error, this key error, and this is because you have a stop codon. If we go up here and look at the protein sequence, it of course ends in a stop codon. And of course, this does not have a mass. So what we need to do is omit the last residue there. And to do that, we can just use a simple indexing scheme. So this 529.translate, that object is a sequential object very similar to a string. It's this BioPython sequence. It follows the same indexing rules as strings. So to fetch all items except for the last, we could use the following indexing scheme here, from 0 up to, but not including, -1. And then if we run it again, we see that we get about 67 kilodaltons as our final mass. In problem 3, write a program that goes through the NGS data and filters out any sequences that do not contain the short motif ACTG. What percentage of reads out of the total starting reads satisfy this criterion of having ACTG in them? So here we have this file, and in the beginning, we're given this block of code here, all records being an empty list and a for loop that goes through the file and spits out record objects. So the iterator in this case will take on the values of sequence records in the NGS data file. And this line here appends them then to this growing list. And what we can do is create a modified version of this piece of code here, which I'm showing down here. And we rename this empty list filtered records, because instead we're going to check for those that have ACTG in them. And instead of looking at the records, instead of appending the records, what we're mainly interested in is the sequence, because that's what we'll need to search for this small motif here. So we have a variable that we just call sequence, and we use this .seek method to fetch the sequence from this record object. And this is something particular to BioPython objects. So this is not necessarily super intuitive. It's not a general Python thing, but for a record object, it has this seek attribute that we can pull out and assign to a sequence. And then we can check if ACTG in that sequence, then append the sequence to our filtered records here. And then to find out the percentage of filtered records, we can just look at the lengths of these two lists that we've created, because the lengths should tell us how many records there were, both in all records and in the filtered records. And if we run this, we see that we get approximately 30.9%. And finally, the optional challenge problem. This problem asks you to consider the T4 phage genome fetched as a DNA sequence below. In it are several protein coding regions. Make a program that scans the DNA and extracts and translates to amino acid sequence any open reading frames, ORFs, signaled by the presence of a start codon, ATG, and one of any possible stop codons, TGA, TAG, or TAA. Include an optional filter that ignores sequences less than 75 nucleotides long. So in this problem, we have, of course, this T4 genome sequence. We run it there. It's composed of DNA bases. We're utilizing a few BioPython modules here. And this is my solution that I'm showing here. So it generates a list of these proteins. You can see all of them end in a stop codon and begin with a start codon, M, which is repeated here for some reason. I guess that is a small error. Let's try running this. Yeah, and so we get 326 entries. Let's see, I think maybe we can fix this methionine thing here. Oh yeah, there we go. So now each one begins with a methionine and ends with a stop codon, and we have 305 open reading frames recognized. And if we just check, we can go to NCBI. We have an open reading frame search. And if we look here for a T4 phage genome, we see that 309 open reading frames are found. So this solution then, I think, is probably fairly close to correct. It's at least similar, quite close to the one produced on NCBI. And it might have to do with the fact that it's looking at a circular phage genome, whereas mine is ignoring the fact that it is circular. But anyway, this is how you could go about doing this. So we have a counter, number of ORFs equals 0, and we're going to go through and try and count the open reading frames. We have a pair of while loops, a nested set of while loops. And we have this counter I here, and this counter is going to indicate our position in the genome. So the first loop asks us to, this first loop is for looping through each base in the genome, each DNA base in the genome. So it will execute everything inside the block so long as this condition is true. And that condition is that I, or our position, should be less than the length of the genome, minus 2. Why minus 2? Well, that is because we're looking at codons, and codons are 3 bases long. So we don't want, when we get to the end, we don't want to be scanning beyond the boundary of the genome when we're looking for our codons. And you can see immediately where we fetch our codon. The way we do it is we fetch the base that's stored at position I, the base that's stored at position I plus 1, the base stored at position I plus 2, and then we add these 3 bases together, right? These are all position. We're using indexes and the positions in the genome to fetch the bases in the genome. And when we add them together, we get a codon. So the first thing we check with that codon then is it ATG? So is it the start codon? And if that is the case, then we begin growing our open reading frame here with an empty sequence. And we have this condition here, stop, which is initially set to false. And this is going to be indicating whether or not we have encountered a stop codon. So of course we just started, so it's false. We haven't encountered a stop codon. But then we're going to scan forward with another while loop. And this while loop will execute everything inside it so long as stop is false and I, our position, is less than or equal to the length of the genome minus 2. So similar to our master loop up there, we don't want to accidentally bump into the end. So these are the two conditions that enable us to search while we're searching in open reading frame mode. And in that mode, we have something called next codon, which is simply the codon using the same recipe that we used to fetch the codon up here. And we'll be tacking it on to yes, we'll be tacking it onto the growing codon. Actually, I'm curious. What happens if we just put codon here? Oh, okay. Never mind. Yeah, so we're growing our next, we're growing our open reading frame by appending to it the next codon. And if the next codon is equal to TGA, TAG, or TAA, then we trigger the stop condition, right? Because these are our stop codons. And if that is the case, then the while loop will stop when we go back into it. And each time we execute this inner while loop, we're incrementing our master counter by 3, because in this case, we're scanning 3 bases at a time. And then we include, of course, so this is basically everything we need to complete an ORF. And then we have another check here, which is simply that checks for whether or not the ORF is greater than or equal to 75 in length. And if that is the case, then it increments the number of ORFs and also prints the translated ORF here. So we're omitting smaller ones. And this is also what NCBI is doing. So this hopefully is why our numbers are similar. And then when we exit this entire loop, this master loop, we simply increment our master counter by 1. So we continue scanning forward 1 base at a time in our master loop, but only scan 3 bases at a time in the inner loop after we found start codon. So that is how the code works. Just run it again and take a look at it. Yeah. So these are, in theory, proteins from the T4 phage. Okay, and that's it for this video. So see you in the next one.